Good morning. My name is Hannah Antosh, and this is Skeletal Muscle Fiber Types for Dr. Sandra Barrera. This is for Biology 515, Human Concepts of Human Anatomy and Physiology 1. Let's go ahead and get started. So to start off, there are three main types of muscles. We have smooth muscle, cardiac muscle, and skeletal muscle. Smooth muscles are going to typically be found within the walls of hollow organs. Contraction of these muscles is typically going to be involuntary, so things such as moving food through the digestive system, right? Moving your intestines, moving food through and then throughout and down and out. Cardiac muscle, and it's called smooth because it's going to typically be found in these flat sheets. Next up, we have cardiac muscle, and this is going to be found exclusively within cardiac organs. Contraction of this particular muscle type pumps blood throughout the body and helps maintain our circulation. Lastly, we have skeletal muscle, and skeletal muscle is going to be attached to the skeletal system. Contraction of this muscle creates movement. This muscle can be responsible for homeostatic as well as non-homeostatic activities. So homeostatic activities could be things such as breathing, chewing, swallowing, shivering when you're cold. Shivering when you're cold, so that fast twitch maintains heat. Skeletal muscles can also be used for non-homeostatic activities such as running, hiking, dancing, typing on a keyboard, activities that we might use for like pleasure. Next up, we have skeletal. So skeletal muscles have several different fiber types. The first type is slow oxidative, also known as type 1. So slow oxidative muscle fibers are for slow contraction. These are going to contain many mitochondria, a large amount of capillaries, a high hemoglobin content, and they're typically going to be red in color. Slow oxidative muscle fibers are typically going to be found in muscles that need to stay contracted for long periods of time without getting exhausted. So think like your lower back and keeping you upright. Think about your thighs when you're walking. Muscles that need to stay contracted consistently without getting overexhausted too quickly. Next up, we have fast oxidative, and these are going to be fast contraction muscles. They're going to contract quickly and decontract quickly. They're also going to have many mitochondria. They're also going to have a high hemoglobin content as well as being red. Finally, we have the fast glycolytic. These also include fast contractions, but conversely, they have few mitochondria, they have few capillaries, they have a low hemoglobin content, and they typically are going to be white in color. The study we're going to look at today was checking how fasting affected the muscle fibers in rats. So for this experiment, they had 38 rats, and they were kept in a laboratory. So they were purchased, they were kept for 1 week for an acclimation period. They had free access to food and water. They were kept at a constant humidity of about 55% and a constant temperature of about 23 degrees Celsius. The rats were old Sprague Dawley rats, and they each weighed about 280 grams plus or minus 20 grams. So after the acclimatization period, the rats were fasted for 96 hours. They still had free access to water, but no access to food. After the fasting was done, blood was collected from the abdominal aorta through exsanguination. They were then dissected, and after dissection, the researchers excised the gastronemesis. That's a hard word, sorry. And the soleus. Those were then weighed and washed with a phosphate buffered saline in order to remove residual blood so as not to skew the weights of them. Six male rats were used for an LCMS analysis and an enzyme-linked immunoabsorbent assay. 8 of the rats were used for a Western blot analysis. 4 of the rats were from the control group, and 4 of them were from the fasting group. For the steroid hormone analysis, the samples were homogenized with an acetonitrile and supernatants, and they were purified. Afterwards, they were processed in the TSQ Quantum Ultra Ultimate 3000. The detection limits of the TS and PGN were about 0.6 to about 14.2 f-moles per milliliter. To test the free amino acids, the rat's blood and tissue were homogenized in an ice cold solution that was 4 parts acetonitrile and 6 parts water. They also added in a 0.1% formic acid into that solution. That was then centrifuged at 8,000 revolutions per minute for about 20 minutes. After that, 1 milliliter of hexane that was saturated with acetonitrate was added. It was then vortexed for 10 seconds and then centrifuged for a second time at 3,000 revolutions per minute for about 5 minutes. After that, it was centrifuged again for the last time at 12,000 revolutions per minute for 5 minutes using a PVDF 0.22 micrometer Ultra MC filter. That filtered sample was then injected into the LC-MS analysis using a Xevo TQD triple stage quadrupole mass spectrometer that was connected to an acuity age class. And that was how they got the samples for the free amino acids. Finally, for the Western blot analysis, the samples were frozen in liquid nitrogen at 25 degrees Celsius until they were ready for use. The solium and the Gastроmenace samples were suspended in an RIPA buffer, then they were centrifuged at 900 revolutions per gram for 10 minutes at 4 degrees Celsius. The protein concentrations were determined using the Pierce 660 nanometer protein assay. The sodium dodecyl sulfate polyacrylamide gel electrophoresis was then used to separate the lysate proteins, which were then transferred into a polyvinylidene difluoride membrane for Western blotting. The membranes were probed with antibodies. They were probed with 2 main antibodies. First up was a mouse monodonal anti-3B HSD antibody. The second antibody was anti-CYP11 beta 1. This second antibody was provided by a Dr. Mokai, and it was raised in a rabbit. So he got that antibody from a rabbit, and then they injected that into the membranes. The bonds were visualized with an Amersham ECL detection system, with an easy capture 2 detection system. The results of this experiment showed that fasting did not significantly change the weight of the solium. I think there was about a 0.02% or 0.02 gram difference in rats that were fasted and rats that were not fasted. The rats that were fasted had an average solium weight of 0.25, whereas rats that were not fasted, rats that were in the control group, had a solium weight of 0.27 grams. So there was not a significant change in the weight of the solium post-fasting. However, in the GM, in the Gastроmenace, the fasting did decrease that fast muscle tissue. So rats that were not fasted had an average weight of 3.2 grams, whereas rats in the control group had an average weight of 4.4 grams. Also, the expression of that 3-beta HSD enzyme that we saw in the antibody section, that was also not affected by fasting. Although the fasting did significantly decrease the mTOR, which is a kinase that plays a role in muscle anabolism, essentially. So all in all, while fasting did have a pretty major impact on the weight of the Gastроmenace, it did not have a significant impact on the weight of the solium, as it only showed a change of about 0.2 grams. fasting has been shown to significantly impact muscle fibers and protein degradation in other studies, just like we saw here. So muscular atrophy, which is the loss of muscle, can be caused through starvation, as we saw in this particular study. However, muscular atrophy can also occur as a result of denervation, heart failure, or even just aging. So, thank you.